Hello, my name is Jonathan Weissman, and I'm a biochemist at UCSF. Today I'd like to talk about methodology that allows us to determine the nucleotide sequence of DNA. And in particular, I'd like to introduce some of the revolutions that are occurring in DNA sequencing that are now starting to make personal genomics a reality. In 1944, Oswald Avery and colleagues did an experiment that arguably has formed the foundation of modern molecular biology. What Avery and his colleagues showed was that DNA was responsible for the transmission of heritable traits, or to put more succinctly, that DNA was acting as the blueprint of life. 9 years later, Watson and Crick solved the structure of the DNA double helix, and in doing so, provided a mechanism by which this DNA blueprint could be replicated every generation. And then about 10 years later, Marshall Nirenberg and Khurana and colleagues solved the genetic code. And what this allowed us to do was to take the sequence of this simple polymer of DNA, A, C, G, and T, and read out the proteins that it was encoding. And these three sets of experiments together really provided the motivation for efforts to determine the sequence of DNA, to do DNA sequencing. And the major technology for about 30 years, from the mid-70s to the mid-2000s, for sequencing it was based on an approach developed by Fred Sanger using something called dideoxy terminators. So what is a dideoxy terminator, and how does this let us sequence DNA? As you know, normal nucleotides have a 5' end and a 3' hydroxyl, and this allows it to polymerize indefinitely into a long chain of DNA. A dideoxy terminator has a proper 5' end, allowing the base to enter to be incorporated into the growing chain. But because the dideoxy nature of it, it's missing a 3' hydroxyl, and that effectively terminates the chain, preventing further polymerization. Sanger then used this trick of having dideoxy terminators as an approach for sequencing DNA. In outline form, the strategy would be you'd start with a template DNA whose sequence you wanted to determine, and then a primer that was complementary to this template DNA. A polymerization reaction carried out by DNA polymerase would then be carried out with a normal complement of nucleotides A, C, G, and T. But one of the nucleotides, in addition to its normal deoxy form, would have a small amount of this dideoxy terminator. So for example, if you put in a dideoxy G at low levels, this polymerization would occur normally, except every time there was a C that was being read out or a G that was being incorporated, there was a finite chance that the dideoxy species would be incorporated, terminating the elongation. So the net reaction is, for the G polymerization, you'd have different length polymers of DNA terminating at wherever there was a C in the template. This could then be repeated with the three other nucleotides, giving you different templates of different length that indicated what the nucleotide sequence was at that position. This was then paired with a method for reading out and for separating the pieces of DNA based on size and reading this out. Sanger was then able to pair this approach of termination with dideoxy nucleotides with a method called polyacrylamide gel electrophoresis that allowed him to separate the different pieces of DNA on the basis of their size. And what Sanger would do then would be to run 4 reactions, the reaction containing the dideoxy A, the dideoxy T, dideoxy G, and dideoxy C next to each other. And in each of these reactions, you would get different size pieces indicating that there was the DNA template at that position had the given nucleotide sequence. So for example, you'd have a small fragment that terminated in C, a larger fragment that terminated also in C, etc. And the beauty of this is by putting these 4 reactions next to each other, you could then read out the nucleotide sequence. So for example, there would be a DNA polymer that ended at C, indicating a C at that position in the template. The next larger piece, one nucleotide larger, terminated at a G, the next one terminated at an A, and then next one larger terminated at a T. This indicated that the sequence was TAGC at this position. And so this could then be repeated over a few hundred nucleotides for each gel, giving the DNA sequence of that template. Here we have an example of a classic polyacrylamide gel electrophoresis system set up to do DNA sequencing. And the heart of it is a long, thin gel here that would then be used to separate the DNA based on size. So one would take the negatively charged DNA, put it at the top here, your 4 samples for each of the reactions, the A, C, G, T, and then close the apparatus, put on electrodes, turn on a strong voltage, a high electric field, and this high electric field would then separate the DNA, pull the negatively charged DNA towards the positively charged electrode at the bottom, with smaller pieces of DNA traveling faster than larger pieces of DNA. And in order to get the kind of separation we need, where we can distinguish one base pair differences in length, we need a really strong voltage. And so it has this nice feature of a safety lock that when the electrode is in, you can't open it up. But basically, it's a fairly simple piece of technology. When you're pouring the gels, which I have to say is quite a bit of a pain and something I don't miss at all, use a high-tech apparatus called the paperclip to hold it together. And you notice there's a lot of urea here, and that's because you use urea to keep the DNA denatured. But essentially, it's a simple 1970s piece of technology, but it works. The next real advance in Sanger-type sequencing was, instead of running 4 reactions and reading out using radioactivity, that it was possible to put the 4 reactions together if one, instead of using a simple readout of radioactivity, a band on a gel or the absence of that band, one labeled the different dideoxynucleotides with different fluorescent colors. So a different color for G, C, A, and T. Now one could run this in a single lane, and by simply looking at the color as a function of position, you could read out the DNA sequence. This then allowed it to be coupled to a novel approach for separating DNA based on size called capillary electrophoresis. Conceptually, capillary electrophoresis was very much like polyacrylamide gel electrophoresis. Basically, you're separating different pieces of the DNA based on their size. But from a practical point of view, it was a much simpler approach. You didn't have to pour these complex gels, and as I'll show you, the capillaries are much smaller than the gels. But basically, the idea is very similar. One takes the mixture of these different length pieces from the dideoxy reaction that are labeled with the 4 different color fluorophores. They're separated then by a capillary based on size, and now you have a laser that excites the DNA as it comes across the capillary, giving you the nucleotide sequence based on its color and size. Here we have a capillary electrophoresis-based system for doing Sanger sequencing. And very much like the gels I talked about, you analyze a dideoxy termination reaction by separating the pieces of DNA based on their size. But instead of looking at radioactivity, we're going to be looking at fluorescence colors indicating the sequence. At the heart of this machine, capillary electrophoresis machine, as its name implies, is a capillary electrophoresis. And these capillaries are, in the same way that a gel was allowed you to separate on the basis of size, a capillary would let you separate DNA on the basis of size. The DNA would enter here, there'd be a strong electric field that would pull it through the capillary, and the time it took to go through this capillary and be detected by the laser, which would be here, would tell you what the size of the DNA is. The one feature of this is you can see these capillaries are much smaller than the gels, and this allows you to do many more in parallel. So for example, this machine had 16 capillaries in it, and that meant that you could do 16 different sequencing reactions at once. And later generations of the machine actually had 96 capillaries, so one could take a 96 reactions like this 8 by 12, 96, and simultaneously sequence all of them. With these approaches in hand, it now became possible to start to sequence simple organisms. So, for example, a virus would have a few thousand up to 100,000 bases, and that really would be readily done by the old gel electrophoresis approaches. Typical bacteria, prokaryote like E. coli, the one living in your colon, has about 5 million bases. Again, it was something that could be done by the gel electrophoresis, but became much easier when capillary electrophoresis got introduced. Similarly with simple eukaryote like Baker's yeast. But where it really became a challenge to get whole genome sequences is when we wanted to start to get genome sequences of complex eukaryotes. For example, you and me. A human has 3 billion base pairs, and it really was essentially impractical to do by gel electrophoresis, and it was actually quite a bit of work to do by capillary electrophoresis. So how was it done? How were we able to determine the genome sequence of humans in the early 2000s? Basically, it was by scaling up. So as you may know, when you want to make a vaccine for influenza, the approach is you take an egg, you put in influenza, and you grow more influenza. And then you take that and infect more and more eggs. And the way we get the H1N1 vaccine is to take warehouses with millions of eggs and just scale everything up. And that's essentially the strategy that was used for the human genome. You would get a factory like this filled with boxes that would run 24/7 and have a computer support and technicians keeping these going. And this really, with a great deal of work, made it possible to sequence the human genome. Just to give you an idea of the scale of this, the Human Genome Project was conducted over 13 years. The cumulative cost of this project was about $300 million. In the beginning of the project, 1990, it was cost something like $10 per base. It would have been an absurdly, untenably high, expensive project, a $30 billion project, sort of like a moonshot type cost. But by 2003, really thanks largely to capillary electrophoresis, that cost had come down to 10 cents a base, allowing the sort of $300 million human genome. Now this really stands as a monument to human achievement, the first human sequence. But of course, not all humans are alike. So there were actually 2 human sequences that came out, one led by a private effort and led by J. Craig Venter, and perhaps not surprisingly, the genome that we got out was Venter's genome. And if you wanted to distinguish, say, a human from a chimpanzee, from an E. coli, one human genome was fine. But of course, we're not all Craig Venter, and we don't all look like him, and we certainly want to know what are the differences in our genomes that, for example, make us susceptible to different diseases. So one genome really wasn't enough. We really wanted, ideally, a genome for every person. And the $300 million cost per genome made that, of course, completely untenable. And so really based on this, people got together and they said, well, how much, if we're going to make sequencing part of the regular medical toolkit, how much is it going to have to cost? And it turns out a high-end, sort of expensive but routine medical procedure, say an MRI, costs in the range of $1,000. And based on this, people put that number, $1,000, on a piece of paper and put that as a challenge and said, would it be possible through advances in technology to make genome sequencing that inexpensive? And of course, it was not going to occur by the influenza model. We weren't going to get more and more warehouses that were going to bring the cost down from $300 million to $1,000. Really had to rethink the way that we sequenced DNA. Now, on the face of it, this challenge seems absurd. It would be as if someone came into a car dealership and said, you know, I don't want to spend $30,000 on your car. I want to spend $1 per car. Can you make cars for $1? Would it be possible? But as it turns out, just sort of stating this challenge provided enormous motivation for different approaches. And this has led, just in the last 3 to 5 years, in an explosion of different technologies that are now, very shortly, will make this $1,000 genome a reality. What I'd like to do is talk about one of those technologies in a little bit of detail to show you where we stand and how it works. And the approach, and as I said, there are multiple ones out there, multiple commercial vendors, but the approach I'm going to talk about, the one that's probably producing the most nucleotide sequence, today is one developed by Illumina and Selecta sequencing technology. And the idea of this approach was really to rethink how sequencing was occurred so that there'd be no separation based on size, but instead sequencing on the slide. And the approach worked by starting with a piece of DNA, what we want to sequence. Actually, you'd start with a very rich pool of DNA. And at the ends of this DNA, you'd put common primers, a 5' primer and a 3' primer. And these primers would be meant to match primers that were physically associated with the slide, allowing the DNA to hybridize onto this slide. This then allowed a localized amplification, generating a cluster of identical DNA molecules. So the single DNA molecule with the primers would land at a spot on this slide and then be amplified to hundreds or thousands of copies. These clusters would then be read out using fluorescent nucleotides. One would come in with a different color A, C, G, and T, and at one cycle at a time, introduce the nucleotide into the DNA, and then use a laser to read out the color of the spot indicating the nucleotide at that position, and then repeat this a second time to get the nucleotide at the second position, and go on repeating it tens or hundreds of times to get the entire sequence of the DNA in the individual cluster. And now what makes this approach so powerful is on a single slide, you could read out the sequence of multiple different clusters just by looking at the color of the spot as you go through the individual cycles of polymerization. And what is really remarkable about this approach is you can get a lot of spots on a single slide. And so in current form, one can get 100 million different spots on this slide. And by following the color of these 100 million different spots through each polymerization cycle, you're in parallel sequencing 100 million different pieces of DNA. It was as if you had 100 million of those boxes in a room working all at once. And as I'll show you, this slide is really quite small, representing an enormous increase in the speed and efficiency with which sequencing can occur. Just to give you an overview of how this might work for, say, a human genome sequencing project, one could take the DNA from an individual, say a patient that you're interested in, fragment their DNA into relatively small pieces, and then each of these fragments would land at a defined position on the slide, be amplified, and then sequenced through this fluorescence approach that I talked about. And then by sequencing, say, 100 million different fragments from that person simultaneously, you can then start to put together in one run a good portion of that person's genome. So here we have an example of a next generation sequencing machine. This one is an Illumina machine, the type I talked about. You can see it's sort of on the same physical scale of the capillary electrophoresis, and maybe naively looks very similar, except perhaps more tubes. The difference is, instead of separating pieces of DNA based on their size, the reactions are carried out, as I diagrammed, on a slide. And this is the actual size of the slide. Just a remarkable thing is that one's able to carry out 100 million different sequencing reactions on this slide. So I showed you before that we had a capillary electrophoresis with 16 different capillary electrophoresis running simultaneously, allowing us to do 16 different reactions, or in the next iteration, 96 different reactions. This is really a completely different scale. Now we're sequencing 100 million at once. And so this machine here actually would let you carry out 1 billion base pairs per day of DNA sequence. In 1, 2.5, or 3 day run, you'd be able to get 3 billion different sequences on the scale of a whole human genome. And that's why they start calling these genome analyzers instead of genetic analyzers, because this is really something that can bring a personal genetics to reality. So as I discussed, their clusters of DNA clusters would be built on this slide, and then a fluorescent nucleotide A, C, G, and T would be put in, and the image of each of the 100 million clusters would be made. And that's what you see here is a computer screen capturing one of these images. I'm sure these dots are barely perceptible, but each of these different spots are a different sequencing reaction. And so you now can imagine how with one small slide we're able to get many, many different sequencing reactions in parallel. And the other aspect of this that's an important point of it is, you'll see down below here, is the computer that makes all of the processing and picking of these individual spots possible. Because obviously you could never by eye pull out the sequences from this spot. Collectively, these new technologies are often referred to as next-gen sequencing, and that basically is a fancy term for these highly parallel approaches for doing sequencing using technologies that are not related to the Sanger sequencing approach. And collectively, this has had a revolution in our ability to collect sequences. So currently, using the Illumina platform, the cost per base pair is 10 to the minus $6, 1/1,000,000th of a dollar per base pair. And the capacity is about 1 billion base pairs per day per machine. By comparison, the most productive capillary electrophoresis machines working at 1 million base pairs a day. In other words, each capillary electrophoresis machine was doing the work of 1/1000th of the Illumina sequencers that I just showed you. And the cost of resequencing a genome today, and today being November 2009, because I say that because it's dropping rapidly, but the cost now is about $30,000. In other words, in going from 300 million to that number of 1,000 that we'd like to reach, we've already gone 10,000-fold of that 10,000-fold reduction, and we're within a factor of 30 or so. And as I said, it's dropping rapidly. I don't think there's anyone who seriously thinks that the goal of achieving the $1,000 genome is not going to be possible. And in fact, it's unlike, say, the moonshot, where Kennedy comes and says, we've got to go to the moon, and 10 years later we go to the moon, but that's it. We haven't gone to the, 40 years later we still haven't gone to Mars. This isn't going to happen with sequencing. It's very likely that we're going to meet the $1,000 genome and go right by it, and it's going to be dropping orders of magnitude cheaper after that. And what this really collectively does is usher in a whole new era of personal genomics. And just two obvious benefits of this will be trying to determine who will benefit or have unacceptable side effects from a given drug. And this hopefully will dramatically lower the cost of developing drugs and make them more effective and allow us to give drugs to the patients who are most likely to benefit from them. The other major advance in this is going to be very helpful in identifying disease genes. So now we can essentially, for any given disease, say Alzheimer's or Parkinson's or autism, we can take affected individuals, resequence their genomes, and ask what about their DNA as a whole is different from people who are not affected by this disease. And when you start to do this, it doesn't just have to be to extreme diseases. It can actually be for any trait. So for example, one of the projects that people are talking about doing and actually carrying out is to take people who are extremely old. So people who have survived with good mental capacity, say to 105 or more, and sequence their DNA and say what is in their DNA that is protective from all the effects of aging that seems to affect all the rest of us so much more dramatically. So what I hope to convince you is that there really has been a revolution in our ability to do genomic sequencing, and that this revolution in turn is going to have an immediate and real impact on human biology and on biological discovery. Thank you.